Perhaps few techniques in biology have stirred as much excitement and controversy as the gene editing technique known as CRISPR-Cas9, usually shortened to simply CRISPR. It allows genetic material to be added, removed, or altered faster, cheaper, more accurately and efficiently than other existing genome editing methods. Here's how it works. CRISPR is actually a technique borrowed from an ancient defense system bacteria used to defend against viruses. The CRISPR complex consists of a strand of RNA bound to an enzyme, usually the Cas9 enzyme, but some groups use a nuclease called CPF1. In the lab, scientists synthesize this RNA sequence called the guide RNA that will determine exactly where the gene editing will take place. Once in the cell's nucleus, the CRISPR-Cas9 or CPF1 complex bumps along the genome, attaching every time it comes across a small sequence called PAM. This allows CRISPR to grab onto the DNA so that the Cas9 or CPF1 enzyme can unzip just a little bit of the DNA double helix. That allows the guide RNA to slip in and sniff around to see if it's a match. If not, CRISPR moves on. But if every base of the guide RNA matches up with the target sequence in the DNA, it triggers the enzyme to cut the DNA in two right where researchers wanted. Now the cell's natural DNA repair mechanisms swing into action. Cells usually repair a break in their DNA by gluing the loose ends back together. That's a sloppy process that often results in mistakes. That may not sound useful, but if adding random bases or stitching ends back together disables or inactivates a disease-causing gene, then that's a desirable result. Just as revolutionary is the ability to insert a desirable strand of DNA into the newly cut double helix. A customized, lab-synthesized DNA sequence is introduced for the cell to use as a template as it repairs and reforms its double helix. Variations on the CRISPR technique can be used to treat genetic diseases, grow more resilient crops, and develop designer foods, drugs, animals, and even malaria-resistant mosquitoes. And more applications are being devised almost daily. But CRISPR can also create off-target mutations with unknown consequences. And editing sperm or eggs to generate designer babies with blue eyes, a high IQ, or Olympic-caliber athletic ability may one day also be possible. Once again, science may be racing ahead of ethics and our understanding of the risks and societal implications of genetic manipulation.